Okay, so I would like to do a few things today. We've got a fair amount of material to cover, and also would like to review the second exam, because remember, your last exam is like semi-cumulative. Okay, so it's very important that you understand these core concepts that we're going over. Um, I'd like to cover 3 or 4 sections. So we're going to talk about the endoplasmic reticulum. We're going to talk about the Golgi apparatus, the uh, lysosomes, and also start getting into cellular metabolism in the mitochondria. Okay. Before we get into the mitochondria, I want to give you guys a refresher into cell metabolism. Um, it's my hopes that you're kind of already at that particular point. I don't really have PowerPoint slides for that. I'm just going to put it up on the board. It's just so that we're all at a place where we can get into some more advanced principles that are covered in this course. Right? And then if we have time, I'd like to go over the lab midterm. Is there anyone in here that's not in the lab? Okay, then we're not going to do that. Or I'll do it at the end if you want to go. I can do that. All right. So the last section was on the nucleus. I posted that lecture. It was just 2 sections. So we did nuclear import and we also did chromatin modeling. Okay, so be sure that you watch that lecture if you haven't done so already. So there's some important principles in there, especially regarding the nuclear pore complex and how proteins get in and out of the nucleus, how mRNA gets out of the nucleus and whatnot. So as we're working through each of these organelles, one thing that is really essential is recognizing what's called the endomembrane system, right? So that's where proteins are formed on ribosomes. They go to the endoplasmic reticulum, and then they have to get processed and travel to wherever their final destination is. So that most often requires that they travel to the Golgi apparatus or then to the plasma membrane. So all of these membranes are interconnected in a sense, and that's why they call it the endomembrane system, right? So membranes within the cell are interconnected and provide a way for proteins to get shuttled to their correct final location, and then also to get fine-tuned and processed. So a majority of that actually occurs in the ER, in the endoplasmic reticulum. And what this section is going to focus on is localization of proteins to the ER. So how proteins get to the ER and then what happens to them there. And something very intricate that's occurring is the membranes that make up the ER are being exploited to position the protein in the correct orientation if it's a transmembrane protein. So when we get into that, it's going to seem a little rough, I think, at first. It's going to all be new material for you. If you've taken biochemistry, maybe some of this was covered in biochemistry, but it's unlikely that it was covered in depth. So just give yourself a break. When you review it, try to review it how I've got it laid out in the PowerPoint slides according to different section titles. That door is open. It should be unlocked. All right, so keep it organized within the chapter. Or within the chapter section. That's going to help you learn it. That's kind of the take-home message there. All right, so what is the ER? The ER is essentially a site of protein synthesis. Now, the true site of protein synthesis, of course, is what? What synthesizes proteins? The ribosomes, right? So, but ribosomes are closely associated with the ER. Now, they are attached to the ER, but they're not part of the ER. Even the rough ER, which is the component of the reticulum that is associated with the ribosomes interact with the ER, but they come off. So they transfer from the cytoplasm to the rough ER and back and forth. All right, but all of these are part of this endomembrane system up here, the ER, the Golgi, how the vesicles transfer from the ER to the Golgi, and we're actually going to omit that whole section. You guys can be very grateful for that. It's a very challenging topic. So be sure you disregard that section. It's not going to be covered on the exam. Very interesting section, but we're not going to get into it. And then also the lysosomes. Lysosomes are an important component of the endomembrane system as well. So their overall role, the ER is generally where the proteins are synthesized, because if the ribosome is associating with it, we say it's being made in the ER. The ER is responsible for folding, some early modifications like the production of disulfide bonds or N-glycosylation. After that, the protein usually will go to the Golgi apparatus for further processing. It's more considerate like fine tuning, and it will finally determine where the final location of the protein is going to be. So it's like a shipping and distribution center of the cell. Sometimes what's produced by the Golgi may need to immediately get degraded. All right, if the cell is overproducing a protein, that's one way of regulating that level of protein. And if it needs to do that, it'll send it to the lysosome. Lysosome has other roles as well, and we're going to talk about those. Very important cellular processes like autophagy and the breakdown of components that are brought into the cell. Okay, so it's a network. The ER is a network of these membranes. The inside of the ER is called the lumen, and it's kind of looks as like these membranous sacs, which we call cisternae, and they extend as part of the nuclear envelope. Okay, so the lumen of the ER is basically the area coalesces with the area between the outer and inner membrane of the nuclear membrane. All right, so about 50% of membranes in the cell come from the ER, or are the ER. So it makes up, it's a majority of the membranes of the cell are actually part of the endoplasmic reticulum. All right, so there's two segregates. There's the rough ER, which is easy to spot under an electron micrograph because it's studded with ribosomes. And those ribosomes are attached, but usually it's temporary attachment, because they can dissociate off and then travel back into the cytoplasm. When a ribosome is in the cytoplasm, we call it a cytosolic or a free-floating ribosome. So the major job of the rough ER is protein folding, processing, and synthesis. The smooth ER lacks ribosomes, hence it's called smooth, and its primary roles are production of new lipids. So if the cell needs new membrane, it's going to acquire it from the smooth ER. It also has an important role in sequestering calcium for the cell. So calcium signaling is a very important component of cell signaling. It's required for muscle contraction as well. So where that calcium is coming from is the smooth ER. Okay, so cytosolic calcium levels are typically low, but if you need to initiate a certain signal, calcium will leak out of the smooth ER and then bind to receptors that are in the cytoplasm of the cell. All right, so there's an electron micro. See the lumen of the ER is the space in between the membranes, and these are all the ribosomes that are interacting with it. And the smooth ER lacks those ribosomes and is the site of lipid and lipid synthesis. All right, so how this endomembrane system was discovered initially, so how proteins can originate in the ER and how they travel to the next part, the Golgi, and then go to the plasma membrane to be secreted to the cell, from the cell, or travel to a lysosome, was performing what are called pulse chase experiments. So it's pretty relatively simple concept. You take cells and you grow them in a medium containing radioactive amino acids. You allow that to occur for a little while right? So the radioactive amino acids start getting incorporated into freshly translated proteins. And then you pull the cells from that medium and place it in fresh medium without any radiation. And then using autoradiography, you can start to see where those radioactive elements are traveling. And they found that it travels along this secretory pathway where things originate in the rough ER, they travel to the Golgi, and they leave the what's called the trans-Golgi network we'll talk about in these secretory vesicles that get released into the environment of the cell. You can see just within a couple hours, you can have proteins produced and outside of the cell. Right? So the initial pulsing is the part where you're exposing the cells to the radioactive amino acids. And the chase is where the cells are incubating in radiation-free serum, radiation-free medium. All right, so now not all proteins are produced on ribosomes that are associating with the ER. So there are many proteins that are synthesized on free ribosomes. And those proteins typically have particular destinations. It makes sense, right? They're floating around in the cytoplasm. So a lot of those proteins are going to go in the cytoplasm, or they're going to go to the mitochondria chloroplasts, or the peroxisomes, right? Some are also translocated to the nucleus. So these proteins are not considered part of the endomembrane system. They're not going, only proteins that are going to the ER are going to be ultimately part of the endomembrane system. Those proteins include ones that are going to be secreted from the cell, ones that are going to remain in the endoplasmic reticulum, or ones that go to the Golgi, the lysosome, or get embedded in the plasma membrane. All right, this is an important concept because as we go through all the different methods of what are called co-translational pathways, you want to keep this in mind. Like we're going to mostly be talking about the proteins that are synthesized on the ER. All right. So what do you think the word co-translational means? Say again. As it's translating, perfect. That's exactly what it means. So this means that as the protein is being spit out of the ribosome, it's actually starting to get targeted to the endoplasmic reticulum. There's a lot of different ways that this can occur, depending on where the localization signal is in the peptide, what the amino acids are like before or after that hydrophobic stretch. And that's what most of this chapter is going to really focus on. There's also post-translational targeting. So these are proteins that are made on free cytosolic ribosomes, and then, like we stated before, are targeted to specific organelles. Okay? So that means the protein gets made, and then other proteins deal with that new protein to shuttle it to the correct location. So we're going to focus first on co-translational targeting. And in order to do co-translational targeting to the endoplasmic reticulum, just like we talked about with proteins getting targeted to the nucleus, obviously there's a signal sequence or a localization sequence involved, right? So the protein, either on its N-terminus, its C-terminus, or actually within the peptide itself, can have a signal sequence that's going to direct it to the ER lumen. Most often, these sequences are then cut off, right? It's not actually part of the final protein. It's just to get it to the ER so it becomes part of the endomembrane system. All right, so this just sums up that previous slide just talked about. So proteins that are made on free cytosolic ribosomes, their fate is going to be the nucleus, the mito, the chloroplast, or peroxisomes. The ones that are performing co-translational targeting, okay, And you can see right there, the ribosome is associating with the ER membrane, and the protein is ultimately getting into the lumen of the ER. This is becoming part of the endomembrane system, right? So they're going to the Golgi. They're going maybe directly to the plasma membrane, or they're going to go from the Golgi to the plasma membrane, the nucleus, lysosomes. So we're going to talk about endosomes today. Endosomes are like a premature lysosome. So you can know that for now, and then we'll talk about the different types of endosomes later. Okay. Thank you for letting me know. I wonder if I'm hitting it accidentally when I touch this thing. Very picky. Okay. So the first scenario we're going to look at, and please let me know if you see that again. Thank you. So the first scenario that we're going to look at is when you have an ER localization signal that comes out on the N terminus of the new nascent peptide. So if you remember from exam 2 content, as the ribosome is producing a protein, what is the first end of the protein that's being made? Is it N term or C term? N term, right? Yeah, so the N term is the first side of the protein as it's coming out of the ribosome, and the C term is the last. All right, so this N-terminal sequence is ultimately going to get cleaved. And what makes this a ER localization signal is basically this stretch of these hydrophobic residues. Okay? A lot of proteins that have to interact with membranes contain these localization signals, these stretches of hydrophobic. Yeah, that's probably right. Let me try moving it over here.
I thought it was on the presentation first started.
It was, but I think you're right. I think it's maybe the space bar. Let's do a little experiment here. Well, okay, whatever. All right, so the way that these localization signals were discovered was it was found that proteins that were made on free-floating ribosomes were a little larger than proteins that ultimately interacted with the ER membrane. And that's because the localization signal is cleaved in the ER. So it makes the peptide a little shorter. And that's what this figure is basically showing. So free-floating ribosomes over there producing the nascent peptide. It's got an N-terminal signal sequence. And those ribosomes, through co-translational targeting, will associate with the ER membrane. And you can see it's actually spitting the new protein in as it's associating with the membrane. One of the last steps in that process is the cleavage of that signal sequence. All right, so what these signal sequences have are basically these stretches of hydrophobic amino acids, right? Now, why do you think that acts as a signal sequence? Think about what's could happen here. Think about what this figure is showing you. So the sequence is going to interact with the membrane. It's going to temporarily get stuck in the membrane of the ER, right? So hydrophobic amino acids associate with phospholipids. Okay, so this is the first mechanism that we're learning. This is the co-translational pathway, right? So that means that you have a signal sequence on the new protein. And with this pathway, there is a particle called the signal recognition particle that interacts with the signal sequence. So you've got a ribosome, it's in the cytoplasm, it's producing a new protein, it's got an N-terminal signal sequence, and the SRP goes and binds to the signal sequence. So when that happens, that protein that's being made is ultimately needed at the ER. So one of the first things that happens is the SRP pauses translation. And that pause is important because if it continued to make protein before it gets to the ER, the protein is not going to get integrated correctly. So that pausing step is very important. Then after the SRP is bound to the signal sequence, it basically brings the ribosome that's paused to the endoplasmic reticulum, and it situates it on top of a structure that's embedded in the ER called a translocon. Right, the translocon around it has these signal recognition particle receptors that the SRP can bind to bring the protein and the ribosome all down and situated correctly on this translocon. All right, so this is what the docking is. The docking event is the ribosome coming and getting onto the translocon. The next part is translocation. That means the movement of the new protein into the ER, and it's passing through this translocon. And then ultimately at the end, the signal, that hydrophobic stretch of amino acids, is cleaved. Now the protein is in the lumen of the ER. Okay, so here you've got a free-floating ribosome. It's doing translation, and it starts to produce a protein. And because the first part out is the N-terminus, and because this protein has the signal sequence on the N-terminus, the signal recognition particle binds to that. It pauses translation, and it brings it to the signal recognition particle receptor, which is closely associated with the translocons. So now this thing is all situated. If you look at the translocon, and we're going to see some 3D crystal images of it, it has a plug. So there's a portion that kind of blocks entry of the peptide into the lumen of the ER. So that will be a definite step that needs to occur where that plug needs to come out of it. But you can see the N-terminal region is now embedded, starting to embed in the lipid bilayer right there. And it moves more and more until it's like completely associated with the lipid bilayer. And now co-translational targeting is occurring, right? So as this peptide is growing, it's actually being pushed down through the translocon. You know, if you think about it, part of the energy that is resulting from translation, right, the actual movement of these, you know, the elongation of this peptide is one of the requirements that needed to push this thing through the translocon. All right, the last step is cleavage of that localization signal, and now you have your peptide inside of the lumen. Okay, now this is important for proteins that are going to either stay in the ER, like in the lumen of the ER, or that are going to be collected into vesicles and sent to the Golgi apparatus. This is not how membrane-associated proteins work. Right? That's going to be the next part that we're going to see. Okay, so if you look at the translocon structure, right, it's a pretty big structure. It's like basically 10 transmembrane proteins that surround and produce this channel. And it has like two main components. Like one of them we talked about is the plug. So this normally seals the channel, and it has to undergo a conformational change to come out so the new protein can actually be pushed through the channel. The other structure is a lateral gate. So on the side of the translocon, there is these alpha helices that can move so that that peptide or that localization signal can transfer into the lipid bilayer. So if you go back here, it's not, this figure is not showing it, the rest of them will. But you can consider like this area right here, the lateral gate, because it's moving out of the way so that the signal sequence can start to travel into the phospholipid bilayer. All right, this gate alternates between closed and sealed, and that's very important in setting up the correct structure of transmembrane proteins. All right, so here you can see the translocon right here. There's the plug on the bottom. And this lateral gate, you can see, is something that opens and closes. And when it opens, it makes like the inside of the translocon accessible to the plasma membrane so that particular sequences like hydrophobic stretches can move out of the translocon and embed itself into the plasma membrane. Okay? Just like is happening right here with the signal sequence. But as we're going to see, it will become more complicated because if the protein is making more hydrophobic stretches, it's going to tell the translocon to open the gate so that that hydrophobic stretch, which is ultimately going to become a transmembrane portion, can move into the membrane. Okay, so just to review, proteins that are destined for secretion or travel to other organelles. These are soluble proteins, right? Not membrane bound. These are the ones that are going to stay in the ER lumen, that are going to go to the Golgi, that are going to go to lysosomes. Now, why do I not have in here on that list proteins that are going to go to the cytoplasm? Very good. Good. That would be produced by a free-floating ribosome. Okay. So, a caveat arises with proteins that need to integrate in membranes. So membrane integration starts in the ER, no matter what membrane it's getting integrated in, unless it's like a mitochondrial membrane. That's produced differently. But if it's in the plasma membrane or the Golgi membrane or a lysosome membrane, the orientation of the protein is set in the endoplasmic reticulum. And there's a reason for that. So the environment in the cytoplasm is the same all throughout the cell, right? The chemistry, the basic chemistry is similar. But the environment inside the lumen of the ER is very different from the cytosol, and it is actually more similar to the environment outside of the cell than it is to the cytosol. Okay? So when proteins are getting positioned on the membrane of the ER so that the N term is in the cytosol or the C term is in the cytosol or the N term is in the lumen, however it's going to be, it does it then so that whatever is in the lumen, if it's going to the plasma membrane, is going to be on the outside of the cell then. Okay? All right, so look here. So here we've got the endoplasmic reticulum, right? So the lumen is in yellow. So this is producing so we had some co-translational targeting that occurred here, and a protein got embedded in the membrane, and you can see the cytoplasmic domain obviously is going to be on the outside of the lumen because now it's in the cytoplasm of the cell. And it gets pinched off of the ER in a vesicle. So the cytoplasmic side is still in the cytoplasm, and the side that's in the lumen is still inside of the vesicle. Now it's going to fuse with like this. This is like the early part of the Golgi, so that the cytoplasmic side is still in the cytoplasm. And as it gets processed through the Golgi, the cytoplasmic side is always in the cytosol. Finally, when it gets to the plasma membrane, right down here, the cytoplasmic side is still in the cytoplasm, right? So whatever is in the lumen of the ER is going to be on the extracellular side for transmembrane proteins that are bound to the plasma membrane. Does that make sense? And it's because the chemistry of the lumen is more similar to the extracellular environment than the cytosol is. Cytosol has special chemistry. It's a highly reducing environment. It's low in calcium, things like that. All right, so you can see these membrane proteins can exist with the N-terminus on the inside or the N-terminus in the cytosol. You can also have transmembrane proteins that have multiple transmembrane domains. All right, so how are these produced? Through co-translational targeting. Well, it all has to do with having these hydrophobic stretches within the protein. Because the hydrophobic stretches tell the translocon to open the lateral gate so that the stretch can start to embed in the membrane. All right, so like I said, the membrane topology, right, is determined here in the ER. Right? The N side is going to be in the lumen, or the C side is going to be in the lumen. That's going to stick and be the same all throughout the endomembrane system. All right, so if a protein has a N-terminal signal sequence, ultimately the N-terminus is going to remain in the lumen. And this is true if there is a hydrophobic stretch within the protein, within the peptide. So just like we saw before in co-translational targeting, you get a signal recognition particle, right? It binds to the signal sequence, it brings the ribosome to the translocon, and the protein starts translating into the lumen of the ER. When it comes across a transmembrane sequence, which is a stretch of hydrophobic amino acids, it causes the lateral gate to open, and then this thing starts moving into the membrane. The N-terminus stays in the lumen, and now the C-terminus, when it's done, is in the cytosol. Okay, so this is for N-terminal signal sequences. Make sense? All right, so some membrane proteins, though, lack an N-terminal signal sequence. Instead, they have an internal hydrophobic transmembrane sequence. Just like the N-terminal signal sequence before acted as a binding site for the SRP, these hydrophobic transmembrane sequences that are inside the peptide can also act as anchors for the SRPs. Right, and then here's where it can get a little tricky. So whether it's going to orient so that the N side is inside the ER or the C is inside or whatever, has to do with charge distribution relative to the transmembrane hydrophobic sequence. All right, it follows this positive inside rule. All right, so here are your two options here. So look at the end product here in both of these figures. So in the top here, we've got the N-terminus in the cytoplasm. And in the bottom here, we've got the C-terminus in the cytoplasm. So both of these have a hydrophobic stretch. You can see here that opens the lateral gate and allows the protein to integrate in the membrane. The thing that's different is in this scenario here, you have positively charged amino acids that precede the hydrophobic region. And in this example down here, you have positively charged amino acids that come after the hydrophobic region. And when I mean before and after, I mean N term to C term. Let's look at this more closely. All right, so here we have a situation where a protein is starting to be made. Here's the N-terminus. And then eventually what comes out is a hydrophobic span, right? So you have a series of amino acids that are hydrophobic, and the signal recognition particle can associate with it. And it brings the ribosome and the newly made protein down to the SRP receptor, and it pushes the transmembrane sequence inside the translocon. The N term is still outside here. The lateral gate opens and the transmembrane sequence travels into the plasma membrane. The protein is continued to be translated until it's done, and the C term of it is inside the lumen of the ER. All right, the reason that this occurred is because there are a series of positively charged amino acids in this region right here. It's keeping, because the cytosol preferably interacts with those positive charges, it keeps it in the cytosol. Okay, so in the other scenario, you have positive charges that follow the hydrophobic region. So here there's going to be positive charges like in this area down here. All right, so the same thing is happening though. The signal recognition particle is binding to the transmembrane region. It's bringing it to the SRP receptor. The transmembrane region is going inside of the translocon and it's getting pushed aside, right? But because the positive charges are here, the C terminus of the protein is staying in the cytoplasm, right? These lack those positive residues, so it's orienting the transmembrane protein so that the N terminus is in the lumen of the ER. Okay, so this is important to understand. It just has to do with relatively where the positively charged amino acids are in respect to that hydrophobic span of amino acids, which is the signaling mechanism to create these transmembrane proteins. Now, why this is important is, like we said, the topology of these transmembrane proteins is determined here in the ER. So when this thing vesicles off, when this protein vesicles off, this is going to remain cytoplasmic. If the target of this protein is the plasma membrane, this is going to be the cytoplasmic side still. This is going to be the extracellular, probably the signaling receptor side. Okay, so now the same rules apply that we just talked about to peptides that have multiple transmembrane sequences. All right, so you're looking at, the positive charges that either preclude or come after all of the transmembrane sequences in the new peptide. But you can have proteins that have multiple hydrophobic stretches, so that what's going to happen is the protein is going to loop in the cytoplasm and in the lumen to produce a more complex 3D structure. All right, so here we've got a internal hydrophobic stretch, right? We've got no N-term signaling sequence like we had initially looked at in co-translational targeting. So the SRP binds to the hydrophobic stretch, it brings it to the receptor, and then you get co-translational targeting. Okay, and the first hydrophobic stretch causes the lateral gate to open, it travels into the membrane. You get continued translation, and then a second transmembrane sequence or hydrophobic stretch is encountered, and it causes the lateral gate to open up again, and that can push into the phospholipid bilayer. All right, so you get continued translation, and look, it comes up on a third transmembrane sequence. That causes it to go through the lateral gate as well. All right, ultimately, the end of this is going to be in the lumen side. Why is that? Why is the C term on the lumen side and the N term on this side? What determined that? Where? Where are the positive charges? Very good. They're right here. Yeah. They might also be located here as well to assist it, but it's that initial processing. Now, if this thing was flipped, and I don't think there's a figure that shows it flipped, there's not, which it should. If it was flipped so that the N term was here, we would see those positive charges here on the C terminal side. Okay. Good. Not that bad, right? Okay, so that's all the co-translational targeting stuff. That's about as bad as all this gets. Now, there is also to create transmembrane proteins, you can also do this post-translational method. I'm not requiring that you guys learn the pathway and the proteins, right? So I took the figured out, but it does it post-translationally, which means the protein is made completely in the cytoplasm, and then proteins interact with it to bring the nascent peptide to the ER, and the ribosome stays in the cytoplasm. Right? This is the post-translational method, and it involves these proteins called GET proteins. Right? So basically, it stands for guided entry of the tail-anchored proteins. But no translocon is involved. No signal recognition particle is involved. Right? The ribosome is not brought to the ER membrane. Just the protein is. This is guided through these proteins, GET proteins, post-translationally. Right, so I have the figure up just so you can see what's happening, but you don't need to learn these proteins. I'm not requiring it. But you can see, unlike co-translational targeting, which would bring this ribosome to a translocon and would involve the signal recognition particle, this peptide is made in the cytoplasm completely, and it's released from the ribosome. And then proteins come and interact with it to bring it to other proteins that are embedded in the bilayer of the ER. Then they cause it to associate in the membrane. Okay. That's all I want you to understand there. You don't need to learn these proteins. Okay, so just to sum up, co-translational targeting depends on the signal recognition particle, whereas post-translational targeting depends on these proteins, these GET proteins. All right. So co-translation targeting, which involves the signal recognition particle, having an internal hydrophobic stretch within the peptide. Both of these occur during translation, right? They're co-translational. But the tail anchored occurs after translation. Co-translational targeting requires the translocon and the signal recognition particle, and of course the SRP receptor if it requires an SRP. And post-translational targeting requires the GET and Sargent proteins. Okay, so that's getting our protein targeted and getting it into the ER or associated with the membrane. But the ER has very unique chemistry in the lumen, so it can provide other post-translational modifications as well. It also ensures correct folding of the protein. Right? So there are molecular chaperones that are very abundant. These are members of heat shock family proteins because their levels greatly go up in response to elevated temperatures. And as that protein is getting pushed into the translocon, these chaperones immediately start binding to it. They keep it semi-linear to prevent protein aggregation and misfolding. All right, so if a protein doesn't fold correctly, it gets targeted for degradation. And that degradation mechanism, it's part of this ERAD pathway, or ER-associated degradation, involves ubiquitin and the proteasome. So basically, the proteins get tagged with ubiquitin right outside the ER, shuttle to the proteasome where they get degraded. All right, protein folding, very important. You get the production of multimeric subunit or multimeric proteins. So if you have multiple subunits that need to get correctly positioned together to produce a final quaternary structure, that's covered in the endoplasmic reticulum as well. Because the ER is an oxidizing environment, as opposed to the reducing cytoplasm, you can produce disulfide bonds in the ER. So the ER is heavy with disulfide isomerases that produce disulfide bonds. So far we've learned about two types of glycosylations, right? So glycosylation is the addition of particular sugar groups onto the protein. we've talked about N-linked and O-linked. N-linked occurs in the ER. N-linked glycosylation is the addition of a sugar group onto a nitrogen. That's why it's called N-linked. O-linked glycosylation is the addition of a sugar group onto an oxygen atom of a specific amino acid. That occurs in the Golgi, and we'll talk about that in a little while. But N-linked glycosylation occurs in the ER. Also, GPI anchors. So GPI anchors are really important in signaling. So it allows cells to get anchored into the membrane, and the addition of these inositol-like anchors are performed in the ER. All right, so here's a molecular chaperone. You can see once this protein is starting to get translocated into the ER, this BIT protein binds. This process does require ATP, but it's very, very important because if these chaperones didn't, the protein would immediately misfold, right? They would also aggregate with other proteins. It would get sticky. We get these sticky complexes inside the ER. So BIT takes the new protein, kind of keeps it relaxed, and then chaperonins or other folding proteins can come in and help it fold correctly. All right, disulfide bond formation. I've said the ER is an oxidizing environment. So when you guys did your SDS page in the lab, remember you treated with beta-mercaptoethanol, right? That's a reducing agent that removes disulfide bonds. So you couldn't create a disulfide bond in the reducing environment of the cytoplasm. You can only create it in the oxidizing environment of the endoplasmic reticulum. All right, of the two types of glycosylations, N-linked glycosylation occurs in the ER. So you get basically this small sugar that gets added to the nitrogen atom of an asparagine residue. O-linked glycosylation is the addition of sugars to an oxygen atom. That's why it's called N-linked and O-linked. And then, of course, the addition of GPI anchors, right? Glycosphatidylinositol anchors. So the way this works, a little unique in that you can see like this is initially starts as a transmembrane protein, and part of the processing involves cleavage of the transmembrane domain and replacement with the inositol lipid. So this is to produce GPI anchors. Now, the way these GPI anchors work, since the anchor is being produced in the lumen of the ER, where is this protein going to sit if it's targeted to the plasma membrane? Is this protein going to sit outside the cell or inside the cell? So this protein here is in the lumen. Remember, the lumen environment is more similar to the extracellular side of the cell. So these GPI anchors have to be produced in the ER in the inner leaflet. So all of these GPI anchored proteins are always going to go to the outside of the cell. Okay, how are we doing? We're almost going to take a break. I'm almost done with this section. All right, so BIT protein is real important in this heat shock, some of these heat shock proteins in preventing protein aggregation and allowing the protein to correctly fold up. But protein folding is a complex process that a lot of randomness is involved. And cells can start to accumulate unfolded or misfolded proteins. So an accumulation of misfolded or unfolded proteins initiates what's called the UPR, or unfolded protein response. And it is a very elegant response that the cell needs to maintain homeostasis. So misfolded proteins can be recognized and immediately pushed out to the cytoplasm, where they're degraded by the ubiquitin proteasome complex. We've learned about that, right? Ubiquitin molecules are stuck on the protein. Then you get polyubiquination by those E3 ubiquitin ligases. And that ubiquitin signal delivers the protein to that large proteasome complex for hydrolysis. Okay. However, if misfolded proteins accumulate inside of the ER, you get kind of some different possibilities that can be done to help the cell alleviate this unfolded protein accumulation. Right? So the ER can actually increase in size, and it can increase the production of chaperones. It can also tell the cell to off itself, right? So it can initiate apoptosis through signaling mechanisms. Remember, apoptosis is programmed cell death, and it's an important component of like tissue sculpting and also preventing cells from becoming cancerous and whatnot. So there are three very elegant sensors for the unfolded protein response. And I do want you to learn these proteins and these pathways. There's the IRE1 pathway to look at. There's the PERK or PERK pathway and ATF6 pathway. So what if you get a buildup of unfolded proteins, how does the cell want to respond to that? Think about it. What are some things that the cell would want to do if too many proteins are accumulating in the lumen of the ER that are incorrectly folded or unfolded? So ultimately, you want to change the transcription that's occurring in the nucleus, right, to maybe increase transcription of chaperones or chaperonins. You might want to increase lipid synthesis to enlarge the ER. Right? Other proteins that are involved in the ERAD pathway, remember ERAD is the expulsion of proteins to get out into the cytoplasm to be targeted with ubiquitin. Okay? So synthesis of those genes is a good response to the unfolded protein response. Right? It's a good way to fix the problem. So there's three ways that it can occur. The first one is this IRE signal. So this is a membrane-bound ER protein, and it's a protease. So when unfolded proteins accumulate in the ER, they interact with IRE1. IRE1 then, as a protease, will cleave this transcription factor called XBP1. Right? It stands for X-box protein 1. When XBP1 is uncleaved, it's inactive. But remember, we've talked about, sometimes protein cleavage is a way of activating proteins, like the insulin protein. XBP1, when it's cleaved, it becomes an active transcription factor that can go to the nucleus and turn on the synthesis of genes that will fix the unfolded protein response, or fix the UPR problem. All right, that's one pathway. Another involves PERK or PERK, phospho-ERK. It's a kinase, right? Kinase is phosphorylate. The target of PERK, which is a transmembrane receptor that interacts with unfolded proteins on one side, is EIF2. So what does EIF2 do? What does it stand for? Learn that one, right? Eukaryotic initiation factor 2, right? So EIF2 is the key molecule involved in creating the initiation complex to do translation, right? So if you phosphorylate EIF2, you inhibit translation. So you get just a global attenuation of production of protein in the cell because we're getting a signal that the cell's sick, right? It's accumulating too many unfolded proteins. All right, the last one is this signal that's bound in the ER called ATF6. So ATF6 is a signal receptor that normally is bound to the chaperone BIP, right? But if you get an accumulation of unfolded proteins, BIP comes off of ATF6 and starts interacting with the unfolded proteins. That activates this receptor complex. All right, so once it's active, ATF6 will move to the Golgi apparatus where it gets cleaved, and one of the active segments or domains of ATF6 is as a transcription factor, so it can go up and initiate transcription. Okay, so these are three very well known pathways involved in protein homeostasis and managing the unfolded protein response. IRE1, PERK, and ATF6. All right, the last part of this chapter has to do with the smooth ER. So I have removed a lot of the material that's in the textbook. We're not going to focus on all that biochemistry, okay? You can focus on that when you take biochemistry. But I do want you to have like an overview understanding of what's occurring. So because membranes are hydrophobic, they can't be produced in a soluble condition in the cytosol. They have to be synthesized associated with existing membranes. And that existing membrane is the smooth ER. So what happens is the outer leaflet of the smooth ER enlarges, right? Because within the cytoplasm of the cell, you have these water-soluble precursors that you need to make new phospholipids. Glycerol 3-phosphate, we know we need that to make a phospholipid, and acyl coenzyme A, we need that to make phospholipids. So I do want you to learn those molecules, but there's a lot of other biosynthesis that's covered in the textbook that I don't want you guys to get into. All right, here's the cytoplasm. Here's the smooth ER. Now, this is just showing you the outer leaflet of the smooth ER, but it's the outer leaflet that is growing. And it's because solubilized in the cytoplasm are the raw materials that you need to produce the new phospholipids of the outer leaflet of the smooth ER. That includes G3-phosphate and acetyl coenzyme A's. Okay, so those basically get synthesized together to produce phosphatidic acid, and then phosphatidic acid can be processed to produce any of those different phospholipids that we learned about back in chapter 2. There was a figure that showed like 8 different phospholipids. There's just different chemical modifications that can produce those. All right, so this presents a problem though. The outer membrane, the outer leaflet of the smooth ER is growing in size, but the inner leaflet is not. So this is going to produce a very irregular and uneven membrane growth. To overcome that, there are enzymes called flipases and scramblases that will translocate newly synthesized phospholipids in the outer leaflet of the smooth ER. You can see how the outer leaflet's growing, and it will move them to the inner leaflet. Okay, it's an ATP requiring step, and this is the only example of phospholipids moving from one leaflet to another that's known in eukaryotic cells. Right, it only occurs here in the smooth ER that's prevalent with these flipase and scramblase enzymes. Okay, so the very last part of this section is basically getting you ready for the next section in the chapter, which we are going to omit. It's pretty intricate, and just for the sake of time, I kind of decided at the beginning of the course that we weren't going to cover it. But this gives you like a brief overview of it, and I do want you to kind of understand what's happening here. And it just has to do with what is the fate of these vesicles that get produced in the rough ER. So obviously they can go to the Golgi. We've talked about that. They can go to the plasma membrane. They can go to the Golgi and then the plasma membrane. They can go to the Golgi and then a lysosome. But before the Golgi, there is this ergic site, which is like a pre-Golgi, you can almost think of it. It's a pre-sorting center where oftentimes proteins are put into vesicles in the ER and shipped out, and they shouldn't be. It's a mistake. And these proteins will carry these specific sequences called KDEL. And it's basically like a reshuttling sequence to get the protein back to the ER. Okay, so you can see here you've got a vesicle produced with the KDEL sequence. It gets into the ER GIC, gets to the Golgi, and then it's sent back to the ER. Okay, other proteins that lack that KDEL sequence will get processed normally. So they'll get to the ER GIC and then ultimately to the Golgi. All right. And that's it for that section. Okay, why don't you guys take a break? I'm gonna, then we'll maybe go over one of the exams. Take 5. But that's right. June. You mind sitting outside while we go? Well, I'll just go over it another day. I'll just go over it. That's okay. That's all right. Don't worry about it. We'll go over it on next week. It's not a problem.
When should I take it?
Soon. How about can you take it tomorrow morning? So I'll be in the lab all morning. Like 9:30. The lab where we're, yeah, I'll be there from like 9:30 to probably about 11:30. So you can come in for 10. Yeah, come in like around, actually come around 10:15 because I have to teach a class in there. And I should have a break around 10:15. Okay. Yep. Also, for the Golgi 11.2 section, there's a lot of information in there. Biosynthesis of all of the glycoproteins. I've taken that out. Okay, again, you'll cover that in biochem. This is not biochem. So I'm not gonna kill you guys with learning that stuff. All right, so be sure you use the PowerPoint slides to help you determine what is on the exam. Okay, so the Golgi apparatus has a funny name, and it is like the distribution center for the cell, because ultimately particles are going to come out of that trans network of the Golgi and get shuttled to the correct location, whether it's the plasma membrane or a lysosome or whatever. It's also a place for fine tuning of proteins, particularly glycosylated proteins. There are many, many enzymes in the Golgi responsible for manipulating sugar groups on proteins to have them function correctly. All right, when you look at the Golgi, it's basically more of these flattened sacs or cisternae, and often you can actually, under electron micrograph, you can see vesicles budding off of the Golgi and into the Golgi. It's a very active organelle. A lot of the biology of this organelle was only elucidated in the last 20, 30 years. It's one of the last organelles in the cell that were really closely looked at. And its importance is really blown up. So it does have polarity in a sense. And what we mean by that is there is a receiving side and there is an exit side, right? So proteins in vesicles coming from the rough ER go into the cis face of the Golgi. It travels through different sections of the Golgi and then ends up on the trans side of the Golgi where it exits. Okay, so cis and trans are not the only regions, though. There are multiple medial compartments which aid in protein modification, specifically those post-translational modifications like glycosylation. And then after the medial, you have the trans region, and then finally the TGN, which is the trans Golgi network, which is the final sorting and distribution center. So the proteins are always moving cis to trans, and often vesicles will recycle back into the Golgi, right? To, so those enzymes can stay associated with the Golgi apparatus, right? So here's the ER down here, up here. Vesicles are coming off of the rough ER, and they're entering the Golgi through the cis face. They travel through the medial region, the trans region, and then finally the last part is the TGN or trans Golgi network, where vesicles are going to either end up on the plasma membrane, they're going to get secreted, they go to endosomes, or they go directly to lysosomes. Right? There are many, many enzymes in the Golgi apparatus that are glycosylating enzymes to form all of these glycoproteins. Right? Now, we talked about N-linked glycosylation that starts in the ER and only in the ER and always in the ER. But the final touches that occur, the fine tuning to those N-linked glycosylated proteins occurs in the Golgi. Right? There are enzymes called glycosidases and glycotransferases or glycosyltransferases that perform further modifications for N-linked glycoproteins. O-linked glycosylation occurs solely in the Golgi apparatus. Right? Remember, O-linked is the attachment of an oligosaccharide to the oxygen atom of either a serine or a threonine, and this occurs specifically in the Golgi. Right? There's about 5 or 6 figures in the chapter that are complex biochemistry figures involving biosynthesis that I pulled out. Okay, so don't get bogged down in those. Most O-linked glycosylated proteins are part of a family called proteoglycans. And proteoglycans are important proteins that are embedded in the plasma membrane and extend out. And they make up a large part of the extracellular matrix. Right? These are proteins like integrins and binding receptors that interact with the microenvironment that surrounds the cell. Right? These proteins are very important in signaling, cell differentiation, cell survival, and growth, because signals that are taken from the microenvironment are going to initiate those changes in the cell to create an appropriate response. All right, so just to review these 2 types of glycosylations, N-linked occurs or is initiated in the endoplasmic reticulum. Right, it's the addition of an oligosaccharide to the nitrogen atom of an asparagine residue. The oligosaccharide, though, is heavily processed and trimmed, and that occurs in the Golgi. Okay. O-linked glycosylation occurs strictly in the Golgi, involves the addition of an oligosaccharide to the oxygen atom of either a serine or a threonine, and it occurs strictly in the Golgi apparatus. These include proteoglycans. All right. Also, there is some lipid metabolism that occurs in the Golgi apparatus, specifically sphingolipids. Remember back from chapter 2. So sphingolipids are important because they make up lipid rafts in the plasma membrane. Remember, lipid rafts are areas of aggregated proteins. And there is an important lipid precursor called ceramide that's produced in the endoplasmic reticulum. And this precursor is essential for these sphingolipids that are important in the aggregation of lipid rafts, also the production of myelin, the myelin sheath, whatnot, and cell signaling. Okay, so most membranes are produced in the smooth ER, but this one specific type of membrane that involves ceramide is produced in the Golgi. All right, so the very last part of the trans region of the Golgi is called the trans-Golgi network, and this is the major sorting and distribution center. So ultimately, vesicles, when they leave here, they know where to go. Where are they going to go? They can go to the plasma membrane, right? And they can go to the plasma membrane for 2 purposes, either for secretion or for membrane insertion. They can go to an endosome, right? So now an endosome is like a premature lysosome, right? Basically, endosomes bud off of the endoplasmic reticulum, right? And they may contain some of the digestive protein that the endosome has. Once that endosome interacts with whatever it needs to degrade, it becomes a lysosome. So there are also what are called recycling endosomes, which are like an indirect route to bring a vesicle from the Golgi to the plasma membrane. And then there's also secretory granules, right? These are going to be targeted for secretion, things like hormones and enzymes. And the other type of endosome is a late endosome. Right? These ones are going to become lysosomes. All right, so look at this figure, just to understand all these different. Now this is a specific receptor, this mannose-6-phosphate receptor. So this is what specifically targets the particle for a lysosome. But so here you have a late endosome that's produced as like an aggregation of all these vesicles that come out of the ER. What makes this into a lysosome, and it's not shown in this picture, it'll be shown in the next section, is something else fusing to this late endosome. And now there's an interaction between the digestive enzymes and whatever target protein needs to be broken down. Okay, so that's one particular fate that can occur. Go to these late endosomes, which become lysosomes. Other things from the trans-Golgi network can be directed for either secretion or integration in the plasma membrane, you can see. Another way of getting proteins to the plasma membrane is through what's called a recycling endosome. So it's just like a vesicle binds to this recycling endosome, and then it can pinch off again and go to the plasma membrane. The trans-Golgi network can also produce secretory granules, which through exocytosis will bind to the plasma membrane and release those particles into the extracellular matrix. Right, so these four pathways are all possible with the trans-Golgi network. Lysosomes are like the digestive sacs of a cell. Okay, their job is to break stuff down. And they do that through having a wide variety of hydrolytic enzymes, right? Enzymes that perform hydrolysis. Remember back from day one, dehydration, synthesis, and hydrolysis. All right, so these things function like as a digestive system as a cell, but there's no absorption that's done, right? It's just chemical or physical breakdown. There's just chemical breakdown. So what are the things that need to be broken down? Well, particles that are endocytosed into the cell, right? Or phagocytosed, so things that come from outside of the cell, nutrients, proteins, bacteria, things like that, bacterial particles. And then there's also particles that originate from within the cell that need to be broken down, such as damaged organelles. So these lysosomes can actually get quite big and quite abundant in cells. And this process of breaking down damaged particles within the cell is called autophagy. And autophagy means, I think it's Latin, might be Greek, I don't know, self-eating, okay? All right, so one thing that's unique about these lysosomes is in order to provide an environment that these hydrolytic enzymes can work, it needs to have a low pH inside the lysosome. So covering these lysozymes are proton pumps, right, that will use active ATPase to pump protons in from the cytoplasm into the lysosome. These lysosomes can take on a variety of shapes and sizes. You can see these guys right here. This is electron microscopy. Yes, yeah, electron microscopy. You can also see mitochondria in here, but variety of shapes and sizes, and very, very abundant in the cell. Right? There's a lot of these hydrolytic enzymes, over about 60, have been well characterized, and they only work at acidic pHs of around 5. And that pH, that proton gradient, is maintained by active ATPase pumps found on the surface of the lysosome. Okay. Many lysosomal storage diseases have been identified as well. So this typically occurs when the lysosome fails and doesn't get rid of old proteins, damaged organelles, and things like that. All right, this is a picture of a lysosome maintaining its hydrogen gradient. Right, so it's actively pumping hydrogens through the use of an ATPase, right, inside the lysosome to decrease the pH, so that the hydrolytic enzymes can actually function. All right, so lysosomes form through fusion of vesicles, right? Vesicles are produced from the ER that contain these hydrolases, right? And then they go to the Golgi, get further processed, and they're released from the trans-Golgi network, and then they start to fuse and accumulate. And the lysosome, what's actually called an endosome at this point, is starting to increase in size. Okay? So these endosomes, right, these early carriers of hydrolytic enzymes, actually act as a really key intersection point between the secretory pathway and the endocytic pathway. Right? Secretory pathway is delivering lysosomal enzymes from the Golgi. The endocytic pathway is bringing materials from outside the cell into the cell. All right, so early endosomes get these endocytic vesicles and they recycle membrane proteins back to the plasma membrane. So this figure is good in summarizing this. You've got two pathways going on here. You've got the secretory pathway. So here we can see, here's the outside of the cell. Right, so the secretory pathway is pushing things to the plasma membrane to get released. The endocytic pathway is pulling particles into the cell to get processed. Right, so particles come in, they associate with an early endosome, and that endosome, as it gets larger and more complex, accumulating all the hydrolytic enzymes and accumulating the particles that it needs to degrade, it then becomes ultimately a lysosome, which you have active degradation occurring in. Okay, so this is showing like a maturation process, right? And think of it, you can't just produce a lysosome at once. It's too big of a particle. So it's produced through the gradual fusion of these vesicles so that it increases in size. And that maturation process goes from an early endosome to a late endosome, ultimately to a lysosome. All right, it's also involved in secretion, though. You get particles that are released from the TGN, okay? And they can get, like we said in the about 3-4 figures ago, it can get sent to these recycling endosomes and put outside the cell, or it can get put into a late endosome. So these endosomes are kind of like a binding point for these two pathways. All right, the process of autophagy is dependent on lysosomes. So like I said, self-eating. And it's a way of breaking down damaged organelles, misfolded proteins, old proteins, things like that. So the other system that we've learned that kind of does this is the ubiquitin proteasome system, but that is very specific for particular proteins, the proteins that have been ubiquinated, right? They've been targeted for death. So it's one protein gets one ubiquitin and then polyubiquination and it's gone. Unlike that method, autophagy is a more general thing. Like you can remove a lot of damaged organelles all at once, and it can remove different things within the lysosome. You might have some old proteins in the lysosome. You might have some beat up mitochondria in the lysosome. You can do it all at once. Okay? So autophagy is a very regulated process. Right? So it begins with vesicles that contain a starter receptor called ATG9. Right? And the vesicles may contain in it something that needs to get degraded, but ATG9 basically directs it to the ER. So this complex is actually going to get assembled on the ER, and then it's going to get released as an autophagosome. And then the autophagosome is going to fuse with the lysosome. And that's what autophagy is. Okay? But there's some key steps in here that kind of mature the autophagosome. So ATG9 vesicles or ATG9 expressing vesicles accumulate on the ER and they start to fuse. And fusion, basically, they're getting larger and larger. And they start bringing in like broken down cytosolic proteins, broken organelles. They form into what's called a phagophore. And then ultimately this thing circularizes right there on the endoplasmic membrane and is released. And now you have an autophagosome. Okay. So they've kind of got it organized into these 4 steps. I mean, it's a very dynamic process. It's just like memorizing these steps and stuff. I don't think there's a lot of use in that, but understanding the process, I think, as I put it together there is important. So why autophagy? So it protects the cell, right? It gets rid of the damaged proteins and organelles, it prevents the accumulation of toxic byproducts and components. It also is a way to recycle nutrients. If the cell is short on amino acids, well, let's go break up some proteins and get amino acids for them. Same with lipids and sugars. It's a good way to regulate energy in the cell. So autophagy is actually coupled to ATP activity in the cell. If ATP or nutrient levels drop, you end up doing more autophagy, so you can produce more raw ingredients. It replenishes the energy stores and the ingredients needed to do metabolism. Okay, so this is a self-survival mechanism, right, to ensure homeostasis. All right, one more section done. Okay, so the next chapter focuses on the organelles, the mitochondria, chloroplasts, and the peroxisomes. Okay, we're going to skip chloroplasts because I'm not a plant guy, even though it doesn't mean it's not important. And it doesn't mean you won't necessarily find it on your standardized exams. But I can tell you, if you're prepping for those, everything that you need to know about the chloroplasts is learned in bio. So not missing much. Right, but we are going to focus on oxidative processes, ones that occur in the mitochondria. Okay, so the very first 2 slides here kind of gives you a general overview of metabolism in the cell. And this is where you really should be at right now, hopefully, with your, having Bio 1 and maybe having biochem or genetics even. Well, genetics doesn't cover any of this, but definitely Bio 1. I'm going to go over this briefly, and then the rest of this section is actually pretty short. It just goes over like mitochondrial dynamics. But if you're weak in this and you're not understanding what I'm going over, before Wednesday next week, you definitely want to revisit this and understand it. So understanding, oxygen-free metabolism that occurs in the cytoplasm and understanding oxygen-involved metabolism that occurs in the mitochondria, right? Because if you don't get this, understanding what we're going to talk about next week is going to be pretty difficult, okay? So what does metabolism start with? Right? It always starts with glucose, right? And glucose has how many sugars? How many carbons on it? It has 6 carbons, right? So in the cytoplasm of the cell, there is a series of reactions that occur that break down that glucose into 2 3-carbon molecules of pyruvic acid. Okay. This process is called glycolysis, which literally means splitting sugar. Okay. Glucose is preferential carbohydrate to get energy from, is broken down in cytoplasm. Right. Now, in order to do glycolysis, there's basically 3 things that need to occur. So one is you need to use a little ATP. And the numbers of these molecules is important because in the end, when we start talking about like the electron transport chain in detail, you're going to want to see, you're going to have to know like how many protons when they move equal how many ATPs. So understanding these numbers, knowing the numbers All right, so in order to do glycolysis, you have to use a little energy. But remember, the goal of this whole thing is to make ATP. And the glycolysis does produce ATP. In fact, for every molecule of sugar, it produces 4 ATP molecules. Okay. So, so that's 1 or 2 of the 3 things. Okay. The third thing are oxidation reduction reactions. So this process right here, going from glucose to pyruvate glycolysis, is essentially an oxidation reaction. Glucose is getting oxidized. Now, if something is getting oxidized, that means something has to be getting reduced as well, right? So the molecule that's getting reduced is the electron carrier NAD+. NAD+ gets reduced into NADH. So in order to do glycolysis, you have to use a little ATP and you have to reduce NAD+. This is the reduction. This is the oxidation. All right, so imagine a cell that doesn't have a mitochondria that can only do this anaerobic form of metabolism. There's no oxygen that's involved. So in order to do anaerobic metabolism, let's say we have the cell and it has an unlimited supply of glucose. What is the rate limiting step for glycolysis? What is going to limit glycolysis? The amount of, the amount of NAD+ that is available in the cytoplasm. So there has to exist something in the cytoplasm that oxidizes NADH into NAD+, and this is called fermentation. So the fermentation pathway utilizes the pyruvate and is broken down, and there's 2 different pathways. Right, there's homolactic acid fermentation, and there's my personal favorite, ethanol fermentation. Right, so if you're a yeast cell, you do ethanol fermentation. If you're a muscle cell, you do lactic acid fermentation. So individuals that are out of shape, right? They their cells, their tissues are not very vascular. Their cells have less mitochondria, which we haven't talked about yet, than individuals that are actively exercising. So when they strenuously exercise, this process is an overdrive to produce as much ATP as possible. When it's an overdrive, fermentation is also an overdrive, and your muscles produce a lot of lactic acid. So when people are out of shape, this goes into overdrive, lactic acid accumulates in the muscles, and you get sore. Okay? All right, so this is the anaerobic process. The aerobic process, when oxygen is present, pyruvate is going to initially get processed a little bit. So there's some processing involved. And ultimately, it's going to be processed into something called acetyl coenzyme. Acetyl coenzyme A in the mitochondria gets fed into the citric acid cycle. So called the Krebs cycle. So the purpose of the Krebs cycle is it produces a very little bit of ATP, right? But mostly what it produces is a heavy amount of electron carrier, NADH, and some other electron carrier called FADH2. That's the goal of the citric acid cycle. Now remember, what is our goal ultimately here? To make ATP, right? So for a eukaryotic cell, making 2 ATP, which is your net, for every molecule of glucose is not closely enough to sustain the life of the cell. You need on the order of something like 30 to 35 ATP per molecule of glucose. So one thing about electrons is that they can fall from higher energy states, right? So like picture a staircase, electron, as it's falling down the staircase, energy is getting released. So collectively, we can take all of those electron carrier molecules, right? And we can start oxidizing them and releasing energy that we can accumulate in the mitochondria. That's what occurs next. Next step is the electron transport chain. Final step is oxidative phosphorylation. Okay. So where is the citric acid cycle occurring? Where in the mitochondria? Good. The matrix of the mitochondria. The mitochondria is the organelle that has an outer membrane, a ribbed up inner membrane, and the region between the outer and inner is called the intermembrane space. And this is the matrix of the mitochondria. So mitochondria has its own genome to supply a lot of the enzymes that are involved in oxidative phosphorylation and the electron transport chain. But in this region right here, you have the citric acid cycle occurring. So you're bringing up, you're generating large amounts of electron carrier molecules, ADH and FADH2. In order to soak up all of that energy that can get retrieved from the electrons, what happens is the electrons get bounced along these molecules, which are called electron carriers, and that energy is used to pump hydrogens from the matrix in the inner membrane space. Okay. So the electron transport chain pumps hydrogens, protons, from the matrix to the inner membrane space. So ultimately, we have a high concentration of hydrogens here and a low concentration of hydrogens here. Because of the chemical makeup, which we're going to learn about today, of the inner membrane, protons cannot leak through the membrane, right? They can actively get pumped to the inner membrane space. The only way they can get back into the matrix is through this enzyme called ATP synthase. So protons can go back in, and when they move through ATP synthase, that molecule is shaped like a hydraulic at a dam, right? Water moves through the hydraulic, it rotates a barrel, and that rotational energy is accumulated and synthesizes ATP. Right? But collectively, all that energy that's transferred from the movement of electrons through activating proton pumps culminates in energy that produces about 32 molecules of ATP just from one glucose. So that's where hopefully you're at. If you're not, that's understandable. If you took Bio 1 a while ago, please review it, all right? Because when we get into the stuff next week, we're going to learn all the steps in glycolysis, and we're going to learn a lot of the enzymes in the citric acid cycle, and we're going to learn how the electron carriers function in the mitochondria. Right? So mito is a double membrane organelle, right? Outer and inner membrane. The foldings on the inner membrane, think about it. Those electron carriers and proton pumps are really the most important molecules. You want to maximize the number of those molecules. So the folding of that inner membrane increases the surface area so that you can maximize the number of those molecules. Also, the ATP synthase complex. In the matrix, you have the citric acid cycle. So all the enzymes that are involved in that geochemical cycle. You have the mitochondrial genome. The mitochondria has its own ribosomes, right? So it does translation in the matrix to produce those enzymes. And then you have the inner membrane space, which is where the protons accumulate. All right, so there's the folded up inner membrane, the outer membrane, the inner membrane space. So the chemistry of the cytoplasm, do you think is more similar to the chemistry of the matrix or the inner membrane space? What do you think? Chemistry of the cytoplasm is more similar to the inner membrane space or the matrix? It's actually the inner membrane space because things can get in and out of that through the outer membrane relatively easily. Right? Molecules are selectively imported, though, into the matrix. Oops. Twice. Okay. You can see here, like things can, if they're smaller than 1,000 Daltons, proteins can move in and out through these pores. But to get something in and out of the matrix is a whole different process and requires signaling. And we'll talk about that later on in the chapter. So what this figure is showing you, these are all the enzymes involved in oxidative phosphorylation and also the electron carrier enzymes that are involved in pumping protons from the matrix to the inner membrane space. Okay. So chemical composition of the inner membrane space is very similar to the cytosol. All right. It acts as a proton reservoir, right? So you get a higher concentration of protons in the inner membrane space as compared to the matrix. And the only way for those to diffuse is back through ATP synthase. So now charged ions don't readily pass through lipids as they do, but you're generating such a gradient in the mitochondria that they could start to leak. So the inner membrane is made up of a special phospholipid called cardiolipin. And the structure of cardiolipin, it really makes it very, very sticky. It has these unusual, it's like two phospholipids stuck together. And these are very abundant in the inner mitochondrial membrane. And it makes the membrane impermeable to protons. All right, so, cells don't just have one mitochondria, right? It just depends on what cell type it is. A muscle cell may have hundreds of mitochondria. A liver cell may have 20 or 50. But the mitochondria are very interactive with each other and very dynamic. So they can move about the cytoplasm and regulate their numbers through two processes. One is fusion, right? And the other is fission. All right, so the fusion is the combining of two mitochondria into one, and the fission, which is very similar to binary fission, is the splitting of one mitochondria into two. The reason it's very similar to binary fission is because these mitochondria were once what? Bacteria. Fusion is mediated by two proteins, MFN1 and OPA1. And it kind of, OPA1 is integrated with the cardiolipin. MFN1 causes two mitochondria to interact. They're found on the outer membrane. And the fusion is important to, mix mitochondrial contents. If one mitochondria is lacking stuff, fuse and kind of fix that. Fission is mediated by this DRIP1 protein that actually causes the formation of a cohesive ring to wrap around the mitochondria and split it. All right, so here's mitochondrial fusion, and here's fission. These things are very dynamic. The cells are always undergoing mitochondrial fission and fusion. These processes are important for distributing the mitochondria as evenly as possible during the cell cycle, right, before mitosis. Right, but you can see during fusion, the outer membranes kind of interact. They bind, the inner membrane breaks down between the MFN1 proteins, and then the inner membranes containing the cardiolipin fuse together, and now you have one mitochondria. The fission event involves this signaling molecule called DRIP1, which when it activates, it forms a cohesive ring that wraps around the mitochondria and pinches it so that it breaks apart. Okay. There's a really cool dye that's available to see mitochondria. It's called MitoTracker. So this is like a HeLa cell that's been stained with MitoTracker and DAPI, which is a nuclear stain. But you can see like the mitochondria don't exist in the cell as we might vision them, like as those bean-like structures. They're actually very highly interconnected and networked through fission and fusion. Okay, that's all we got today. Like I said, if you're kind of weak on metabolism, try to warm up a little bit before next week. The book will provide everything you need to do that. But if you need a more thorough explanation, maybe pull out a Bio 1 book or something, or YouTube will do it too. Okay. For the sake of time, what we're going to do is not go over any exams today because this took a lot longer than I thought. We'll go over the lab exam in lab. So go ahead and just keep that and bring it to lab on Monday. And then we'll go over exam 2 next week after everyone's taken it. All right. Hope you guys have a wonderful week. I will see you on Monday. Yes, sir.
Oh, I just need my lab exam. I don't think I got it.
Yes, ma'am. Yeah, so you did well on this. So, yeah, it was curved. This is your uncurved score.
Oh, really? Thank you.
Yeah, that score that's on your midterm is your uncurved score. All right, so your grades were curved.
Thank you.
You're welcome. The grade that's on Canvas is your final grade. Okay. Remember, suit up and show up to the lab. That's really what I mostly care about. Lab.
Look at that again right now. I'll look at it.
Fry, your little fry.
Yeah.
So, yeah, so I promise you, you look at that a couple times, it'll start to make sense. Remember, the stuff needs to move in and out to get recycled. I wouldn't think of it like that, but just think of it as a spatial thing, right? So, I'm not looking at the figure. I'm just trying to remember the importers and the exporters, right? The importer is going to bind to the proteins and bring it in. That means that importer protein needs to get back out. And it does that through the GTP switch. So this that RAN protein sits there on top of the nuclear pore to, hydrolyze GTP to move that thing in and out. But why don't you look it over, and then we can get together whenever you want, either by Teams or next week on Monday, whenever you want to do it. I can go over it with you again. It's a tricky one. Yeah, man. All right.